Professor Leon Glass is from Canada, and he is a Royal Fellow of Royal Society of Canada and American Physics Society and the American Association of Advancement of Science. He has won many prizes and fellowships. His current focus is on the application of mathematics to study biological functions reasons in the cardiovascular system, dynamics and control in genetics and neural networks and visual perception. And he has co-authored a book, and it is in Chinese. You can find that. Yeah. Today's talk title is Geometric Aspects of Dynamics and Diagnosis of Cardiac Arrhythmias. This talk is an interesting talk. Actually, I got a problem before. My heart beat very fast.
Oh, okay.
That's okay. Yeah, it's very common.
Okay.
Thank you very much for this invitation, and it's really a privilege to have the opportunity to visit your city. It's the first time I've been in Hong Kong, and I thank you all very much for coming. So there was a little bit of confusion concerning the title. There are really two titles. I'd like to talk a little bit about the broader picture of something that I call dynamical disease, with particular applications of this to cardiac arrhythmias. Now, I have had a contract and a license agreement with Medtronic. I'll tell you a teeny bit about that in a second. And also, I am not a medical doctor. I'm curious to know if there are any people here who are medical doctors. Okay, then I can say whatever I want. Okay, I'll give you some background and history. I'll talk about what I call dynamical disease with cardiac examples, and then I'll talk a little bit about cancer and evolution. So most of my research is associated with problems related to cardiac system, and the work, the discussion concerning cancer and evolution is much, much more speculative, and it partially arose out of a conference that was held in Barcelona last year, organized by Professor Smale also, at which I participated. Now, I start this discussion back in Aspen, Colorado in 1975, when there was a meeting at the Aspen Center for Physics where Professor Smale was giving a talk, and there were many other people who were there. This is some comments by John Guggenheimer, in which he points out that Professor Smale gave a talk related to iterations of this simple quadratic map. This was talking about possibilities of chaos in this system. This was very influential for people who were at the meeting, in particular Mitchell Feigenbaum was there, and he later contributed to analytic insights into the properties of this system. This is mentioned Albert Liebschaber, who did physics experiments related to this. John doesn't mention me, but I was also there, very excited, because in the summer of 1975, I was moving to McGill University, and before that, I had always been in physics departments or physical science departments, and based on this discussion and talk, I was very interested to see if chaotic dynamics and interesting nonlinear phenomena could be found in biological systems and physiological systems, and whether they were relevant to the health. When I got to McGill, there was a colleague there named Michael Mackey. Michael Mackey had training in biophysics. He was already a professor at McGill University. The picture there is how he looked about that time. There was just a symposium at McGill University because we both turned 75 in the past year. So this was in honor of that. And I was asking Michael whether or not he knew of examples of chaotic dynamics in physiological systems. And he said no, but we started to investigate and started to do research in that problem. The paper that we worked on at that time proposed a time delay differential equation as a mathematical model of a physiological system. The physiological system was a system for control of the production of blood cells. The basic idea behind that equation was the observation that if you're very sick, your body cannot produce a lot of blood cells. If you have a lot of blood cells in your body, then you don't need any more. So if you try to study the production rate of blood cells as a function of the number of blood cells in your body, there would be a maximum at some intermediate value of blood cells, not too large, not too small. But in the body also, there would be time delays between the time the body got the signal to generate blood cells and the time you would actually manufacture them a day or two later. So in order to describe this physiological system, we proposed a time delay system, time delay equation, and this equation displayed chaotic dynamics for certain ranges of parameters which we studied in that particular paper. So in at least the model system, we were able to find the possibility for chaotic dynamics. Now, in this slide on the bottom shows dynamics that were present in that system, and up above it, it shows the sorts of dynamics that people can measure if they take your blood samples on a regular basis. At the end of that paper, we proposed that there are a class of dynamical diseases. Dynamical diseases, the notion was that dynamical diseases would have interesting dynamical properties that would have an onset associated with a bifurcation in the dynamics in some associated mathematical model. So the physicians often recognize the presence of diseases by noticing qualitative changes in the dynamics. So we were saying that the correspondence of that in mathematics are equations describing those systems which would have qualitative changes in the dynamics which we wanted to study. So we laid out ways this could be studied, in which we said one could demonstrate the onset of abnormal dynamics in animal models of the diseases by changing control parameters. One could gather detailed experimental and clinical data to determine what bifurcations were taking place in the real biological or medical systems. And we could also attempt to devise therapies for disease by modifying and manipulating the control parameters back into some normal range. We did not say anything about the possibility of predicting what dynamics would happen in the future, but this is something we should have said, because it's also another way in which understanding some aspects of mathematics can be applied to make sense of biological systems. Now, in analyzing dynamical disease, we can use dynamics to aid diagnosis. We can develop mathematical and biological models. We can develop therapies. I would say that in terms of what has been done mathematically in this area, this is still something which is really needed. There's not been very much practical work in terms of developing therapies yet. I won't say very much about that. And we can also predict imminent transitions. Once again, the basic idea is that instabilities in dynamical system lead to qualitative changes in behavior bifurcations corresponding to changes in dynamics that are phenomenological, that are macroscopic, that are often observed medically, leading to diagnosis of disease. In the talk today, I'll talk about cardiac examples, several different cardiac examples, and then towards the end I'll talk about growth instabilities in cancer and karyotypic evolution in cancer, trying to make some contact with some of the other talks that have taken place in this particular series. Okay, so this is a picture of the conduction system of the heart. The normal rhythm in the heart is generated by the sinoatrial node, a little region of muscle cells, and this sends off waves this sends off waves of activity that go over the atria, down into the ventricles, leading to contraction of the heart, pumping of the blood to the body. Now, this is an electrocardiogram, which all the older people in the audience have had these taken. So next time you have it taken, you can ask to see it. These big boxes represent 200 milliseconds, a fifth of a second. The shapes of the complexes on this give physicians an idea of what's happening inside the heart. The electrocardiogram is a display of the potential difference on different points, between different points on the surface of the body. The P wave is this bump is associated with the excitation of the atria, the upper chambers of the heart. The QRS complex is associated with the contraction of the atria and the excitation of the atria. The T wave is associated with the recharging of the atria, the termination of the heartbeat, and getting ready for the next heartbeat. The typical time between heartbeats is 800 milliseconds to 1.2 seconds. These abnormal complexes are called ventricular premature complexes, and they're easily distinguishable, even if you're not a cardiologist, by the abnormal shape. Okay? So this is a normal electrocardiogram with two abnormal complexes, and almost everybody in the room will have abnormal complexes at some point in their lives. So if you know that you have these, don't be very worried by what I'm going to say later. Okay? This is an example of a rhythm that's called atrial fibrillation. This is a rhythm in which the upper chambers of the heart, the atria, are no longer being driven by the regular pacemaker of the heart, but there's irregular activity in the atria. The irregular activity leads to irregular activity in the vent, going and exciting the ventricles, and this very irregular frequency of the heartbeat. So each one of these now corresponds to a heartbeat, but the rhythm tends to be fast. It increases, the likelihood of it increases in elderly people. It is generally not fatal, but it reduces the ability for strenuous exercise, and it can lead to strokes. So it's a rhythm which is very often treated by medical doctors because it doesn't kill you if you get it. So as people get older, there's increased prevalence. Now, we were made aware that there was a problem associated with doing automatic diagnosis of atrial fibrillation. Because of the irregularity, you might think that analysis of the standard deviation of the interbeat intervals may enable you to identify this, but that in fact was not particularly good. what we propose to do, and this is with postdoctoral fellow Katsumi Tateno, is to analyze the histograms of the changes in the intervals between the heartbeats. So we looked at the probability density function of the changes in the intervals between the heartbeats. We filed a patent. That patent was licensed to Medtronic. And ideas behind the patent were incorporated in a device that Medtronic has, initially called the Reveal XT, and now the Reveal LINQ, which is able to do automatic analysis and give the physician information about what percentage of time the patients will be in atrial fibrillation. This is something which can be implanted under the skin and can be downloaded. So it evidently is quite useful to physicians. This was very gratifying because the notion that mathematics and mathematical approach can lead to useful results is gratifying, but this does not use very sophisticated mathematical ideas to analyze it. With my colleague Alvin Schreier, who I've been really blessed to collaborate with for, I guess, over 40 years, we've studied an experimental system, and let's see if that's going to work. Okay. This is, can we see that? There. It's spontaneous beatings. It's just a few beats, spontaneous beatings of this little ball of cells. So this is from chick heart, embryonic chick heart. It's about 1/10 of a millimeter in diameter. It beats spontaneously, and it can be studied by sticking microelectrodes into it. Those microelectrodes can be used to do a variety of experiments. It's possible to record the activity. It's possible to stimulate the cells and see what happens following single or multiple stimulation of the cells. Following a single stimulus, the timing of the subsequent beats will be reset. They will occur at a different time than what would happen if you had not given the stimulus. That type of experiment is used to determine what's called a phase resetting curve, and the phase resetting curve gives the effect of the pulse on the timing of subsequent beats as a function of the phase of the pulse. One can use the experimentally measured phase resetting curve to predict the effects of periodic pulses. The notion is that the heartbeat is generated by a stable limit cycle oscillator, that the stimulus takes you from one phase of the oscillator to another phase of the oscillator, and then following that resetting, you wait a time, and then you give another stimulus. So the equation to represent this is a map. It's a difference equation. It's a nonlinear difference equation. And it's a nonlinear difference equation that can be experimentally measured. But it's a nonlinear difference equation which represents the resetting of one phase on the circle to another phase of the circle. Such equations are called circle maps. So to understand the mathematics of this, one has to understand something about the properties of circle maps, and analysis of those problems goes back to Poincaré, but then exploited particularly by Arnold and others subsequently. Now, the observation in this particular system is that depending on the frequency and amplitude of the stimulus, one can get regular rhythms in which for each stimulus, for example, there's one beat, or one could also have irregular rhythms. So this example is an example that we associate with chaos. These spikes are the stimuli. The complexes before the spike is what's called an action potential associated with the contraction of those little aggregates. One can plot the phase of the stimulus, the time from the beginning of this action potential to the spike, as a function of the phase of the preceding stimulus. For the data here, for a longer segment of which this is excerpted, one has that plot being a non-monotonic plot, which is similar to the types of functions which we know can give rise to chaotic dynamics in these systems. So do people know who that is? Huh? Okay, that's some of the people here are old enough to remember this. Young people, it's from Jurassic Park. It's Jeff Goldblum, and if you don't remember what chaos is, go to YouTube and you can see Jeff Goldblum explain what chaos is. What chaos is associated with aperiodic dynamics in deterministic systems. Aperiodic dynamics in deterministic systems with sensitive dependence to initial conditions. So this has been something which we have been interested in. Okay, now we can also use these experiments, use this model system to predict, same system, to predict dynamic transitions. A previous postdoctoral fellow, Min-Young Kim, who was visiting from Korea, studied the effect of adding drugs to these aggregates as they were beating regularly. So she added drugs which would block a particular channel, a potassium channel in these aggregates. Under addition of the drugs, this is just now experimental data. This axis is the time between the heartbeats. This is now in this model system. This is time as time evolves. The interval decreases, and then the interval actually started alternating back and forth between a short value and a long value. So this is an example in this situation. This is the sorts of pictures that one often shows in mathematical models of chaotic systems in which one plots a bifurcation diagram. This is an experimental system in which the system is generating the bifurcation diagram as the concentration of the drug changes as it diffuses and affects the properties of this experimental situation. A graduate student, Thomas Quayle, observing these rhythms, said, well, possibly we would be able to predict when this alternation is taking place, or that it will take place before it takes place. Now, I will point out, and we could discuss this if people are interested, that in people, there is some evidence that before the onset of a dangerous or fatal rhythm, there are alternating rhythms taking place. So, from a point of view of medicine, if one could predict potentially fatal rhythms before they occur, that would be an advantage. So, in this model system, the attempt was made to predict those transitions before they occurred. Now, this is just this similar phenomena from a different experiment. In mathematical studies of bifurcations, it's known that as one approaches the bifurcation point, for example, for one of these period doubling bifurcations, that the time to get back to either a fixed point or to a cycle increases in the neighborhood of that bifurcation point. So if we give a perturbation, if we're changing a parameter, and here we have a stable period 1 cycle, and then we give a perturbation, we can come back to that over a period of time. As we get closer to the point where that bifurcates to a period 2 cycle, the length of time that it takes to get back to that increases. The fluctuations will increase in a noisy system, as the correlations will increase. So in the experiments, one can plot, and those concepts have been called critical slowing, and they're well known in the physics literature. One of the early papers from that was done by Hao Bai Lin, who was a professor in Beijing. So what Thomas Quayle did was developed a technique to exploit those increased fluctuations prior to the bifurcation. He came up with a criterion that was applied in several different experiments to try to predict whether or not there would be a period doubling bifurcation occur. And it turned out that worked, but the length of time from the signal to the time when there was actually that bifurcation in the doubling of period was very variable in the different experiments. Now, this is the type of data that I find amazing and challenging and fascinating. So this is an electrocardiogram from a woman who died at the age of 82 years old while she was having her electrocardiogram being recorded.
So it's recording and then she died?
Hmm?
So it's recording the electrocardiogram and then she died?
Yeah.
Wow.
Yeah. At 3:42 in the afternoon. Yeah, so people feel free to ask questions. Thank you for asking.
So it's basically witnessing her death, right?
That's it.
Okay.
Yeah. These are normal heartbeats. These are premature ventricular complexes, like I described before. At 4 in the afternoon, there was a normal beat of premature ventricular complexes. These alternated, and then there was this little rhythm of multiple premature ventricular complexes, which is called by a cardiologist non-sustained ventricular tachycardia. Ventricular tachycardia is a fast heartbeat coming from the ventricles. It's called non-sustained because it spontaneously started and stopped. After that, in this interval, things were kind of quiet, looking more like this than that. And then at 7:15 in the evening, there was this alternating rhythm once again. There was a sequence of non-sustained ventricular tachycardia. There was a single normal beat. And then there was this ventricular tachycardia, which was a fatal rhythm in this lady. Now, to me, it's amazing that our hearts can continue, or this person's hearts continued successfully for 82 years. And at this particular point in time, something happened that led to the initiation of a rhythm, which was a fatal rhythm. Now, when one looks at records like this, you have to think that these abnormal heartbeats are playing some kind of role in what's happening. The first beat here is very similar to the beats that are happening that don't lead to anything happening. So if one looks at records like this, it seems as though these abnormal beats are playing a role. That occurred to cardiologists. What cardiologists did is that they have drugs that can reduce the number of abnormal beats. So they have patients, in this case they took 1,500 patients, all of whom had had a heart attack, a blockage of the coronary artery, all of whom could take the drug, and the drugs reduced the number of the abnormal beats. Okay, the drugs reduced the number of abnormal beats. 750 took the drug, 750 did not take the drug. They count the number of people who die as a function of time, and the people who took the drug were more likely to die as a function of time than the ones who didn't take the drug. So they had a hypothesis that the drug would help reduce sudden cardiac death. They refuted the hypothesis. This is back in the end of the 1980s. As a result of this, cardiologists now tend not to be very interested in these extra heartbeats. Most of the heartbeats, extra heartbeats, as I said before, happen in normal people. Most of them are benign. But it's also known, part of the cardiac literature, that an increased incidence on a population basis, increased incidence is associated with a higher risk of sudden death. Okay? So we also believe that they're benign in some people, but therefore they're not benign in others. They have significance in others. I'm fascinated by understanding something about the dynamics of those extra beats. So this is a record. I don't know if you folks can see this. The front rows can see it. So this is a record. So one of the things you can do if you're not a cardiologist is you can count the number of normal beats between abnormal beats over time. Okay, so you can look and you can say how many normal beats are there between each two abnormal beats. And this is a sequence of integers. So those are a sequence of integers counting the number of normal beats between abnormal beats as a function of time. Notice that down here, this is in one person, not the other record, one person over 24 hours. Notice that some of these numbers are, 1,985. Like a half an hour goes by with no abnormal beats. Other numbers are 2s and 3s. Notice over here, there's some red numbers over there. Over the course of some reasonably long period of time, like a half an hour or an hour, this person's heart was generating odd numbers for the number of normal beats between abnormal beats. So all the numbers in that sequence of the numbers of normal beats between abnormal beats were odd over a long period of time. I think that's amazing. Okay? We're interested in trying to, and I would also say that from a point of view of medicine, data such as this is not looked at, and it's not, whether it's significant or not is not known. Now, in order to, so this is a record. If there was a physician in the room, we would ask the physician to analyze the record. This is a record from a 16-year-old boy who had fainted when he was in the cafeteria. And he came to the cardiologist, and this was what the electrocardiogram looked like. So these are normal beats. He had had surgery on his heart when he was a child. These are normal beats. These are the abnormal premature ventricular complexes. So if there was a cardiologist in the room, we would ask the cardiologist to analyze what was happening. However, I was just at a meeting. I came here from a meeting in California where there was a cardiologist in the room, and he did not do a good job analyzing it. Okay? A mathematician might be able to do a better job. This rhythm is almost certainly generated by a mechanism in which there are two pacemakers, each with its own fixed rhythm, competing for control of the heart. One of those rhythms is the normal sinus rhythm that I mentioned at the beginning. The other rhythm is a rhythm that's set by the ectopic pacemaker. There's another pacemaker which is generating these abnormal beats. That's our interpretation of this rhythm. Here's the normal rhythm. Here's the abnormal rhythm. If the abnormal rhythm falls during a short period of time after the normal rhythm, then it's blocked, and you don't see evidence of that abnormal rhythm on the electrocardiogram. If that abnormal falls outside of that time, this is called the refractory time. If it falls outside, you see the abnormal rhythm, and the next normal beat is blocked. This is just the properties of the heart. If they fall at exactly the same time, or almost exactly the same time, then you see a complex which has a different morphology than the usual normal morphology, because the excitation is coming from a different way than it does during the normal heartbeat. So here you have one rhythm going along, normal rhythm. You have the abnormal rhythm going on a little slower, and there's this competition between what happens in those two rhythms. If we look at this, we can count the number of normal beats between two abnormal beats. Here we have 1, 2, 3, 4, 1, 2, and 1. So in this particular trace, we have either 4 or 2 or 1 normal beats between the abnormal beats. So we can ask a question, can we understand that mathematically? 1, 2, and 4. Can we have a theory for those numbers? So the answer is yes, because this mechanism is very simple. In fact, the problem is exactly equivalent to a problem that was studied in number theory called the gaps and steps problem. The problem is not very well known in number theory, but there was a paper by somebody called Noel B. Slater back in the 60s, which analyzes problems of this sort. In the current case, for this mechanism, two independent rhythms, one sinus cycle time, one ectopic cycle time, one refractory time. There are three parameters, all that have the units of time. So you can normalize. You're in a state of two parameters. In that state of two parameters, we can block up the plane to say what are the numbers of normal beats between abnormal beats that would be observed according to this mechanism. And that's shown in the following slide without going into, so each one of these regions, including the ones not shown, has regions in which those triplets of numbers occur. Those triplets of numbers, so what happens is for any value of those three parameters, in general, there will be 3 normal beats between every two there'll be 3 different values for the number of normal beats between 2 abnormal beats. 1, 2, and 4 are 3 different numbers. One of those numbers is odd. 1 is an odd number, 2 and 4 are even. And the sum of the 2 smaller ones is 1 less than the larger one. So 1 plus 2 is equal to 3, which is 1 less than 4. In this region over here, this is now the refractory time divided by the sinus cycle time. This is the abnormal cycle length divided by the sinus cycle length. And from measurement in that particular record, we find ourselves in this particular region over here. Now, one of the things that we have been interested in doing is trying to develop ways in which one can summarize the dynamics that take place over 24-hour periods in which there are many frequent extra beats happening, and figure some graphical way in which we can summarize the dynamics that we can see over this long period of time. So we've developed something, and I will show it, and I'll just briefly talk about it, something we called a heartprint. This axis is the time between the normal beats. These are kind of heat plots or density plots showing histograms of measured values. Let me just talk about the coupling interval in this. The coupling interval is the time from the normal beat to the abnormal beat. This is readily measured on the electrocardiogram. In some situations, for example, for this particular patient, this histogram, the coupling interval is not focused around single values. It's over a broad range of values, consistent actually with the mechanism that I just described before, where there are the two independent rhythms. In other patients, however, this coupling interval tends to be very focused and very sharp around particular values. This indicates that there is a different mechanism in different people generating these abnormal beats. I also believe that if we knew better how to interpret what's happening, we would be able to come up and understand more about the mechanisms than is currently understood, based on looking at the phenomenological properties that can be measured readily. I'll just point out that a particular genetic defect, so this series of talks is associated with genomics, particular genetic defect in patients, a defect in a potassium channel, leads to lots of frequent extra beats in which there is tens. In this group of patients, these are collected by colleagues in Mexico, Marquez and Lerma. This data is not published. There tends to be sharpness in this coupling interval of these extra beats. And in one study, we showed that there was some correlation of the sharpness of this interval with a higher risk of sudden death. This is just a correlation. It's not strong enough to make clinical decisions, but in my mind, it indicates that there's the potential that understanding these properties may be particularly interesting. Now, okay, I'll try to do this next section in a few minutes. So in the normal heartbeat, there's a little range of cells, which is a pacemaker, which sends off regular rhythms, which set the heartbeat. Or regular, there's also natural fluctuations of those rhythms. In abnormal rhythms, one of the mechanisms for abnormal rhythms says that instead of the rhythm coming from a pacemaker, the rhythm is coming from an excitation which is traveling in some sort of reentrant loop. Okay? So it's not set by a pacemaker, but it's set by a rhythm in a loop. This is a picture from a cardiologist resetting an entrainment of ventricular tachycardia. The cardiologist was Mark Josephson, who died in the last couple of years. He was doing exactly the same types of experiments on patients who had tachycardia, who came to him, who were referred to him for medical treatment, as we were doing on the chick heart cells in the laboratory, resetting and entrainment. This is not a picture of Mark Josephson, but a cardiologist. He has a catheter. The catheter is being inserted in the femoral vein of the patient. It's being pushed into the heart, and through the catheter, he can do three things. He can record the local activity. He can do electrical stimulation and record the activity, and he can deliver radio wave excitation, which, just like your microwave, heats up a local region of the heart, it burns it actually, and kills a local region of the heart. So what that cardiologist is trying to do these studies in which he does stimulation at different phases of the cycle, at different frequencies, just the same types of experiments we were doing in the chick heart, but he has a purpose in mind, and his purpose in the mind is to determine where to put a burn to solve the arrhythmia. Now his picture of what's happening is the excitation is traveling around a ring like this. If the excitation is traveling around a ring like that, then how is he going to cure? Okay, so here somebody has to say something. He's going to burn something to cure the rhythm. So I would claim he's thinking like a topologist rather than a doctor to cure the rhythm. What's he doing? How does he cure the rhythm? Do you understand the question? How can burning the heart, he's destroying part of the heart. How can that cure the rhythm? That's right. He's cutting the ring. Okay? He's changing the topology of the circuit from a ring to something that's not a ring. That's literally what he's trying to do. That's what they do. In order to study these problems, we have carried out experiments, and these experiments were initiated initially by Gil Bubb in our laboratory, and there's a recent paper in which we did in the journal Chaos, which describes what happens when we now take these chicken heart cells and grow them in the shape of a ring. So here's a ring. The excitation is traveling around the ring. And what this slide shows is that in different experiments, we can get different geometries of excitation. So this is measurement of excitation from calcium waves. The ring, the tissue in here is cells from chick heart once again, and this is about 1 centimeter in diameter, and the ring is just a couple of millimeters wide. So here we have 2 rings going around. Here we have a little spiral wave going around, sending off waves that collide and block when they reach there. Here we have a pacemaker sending off waves that collide and block. Here we have another pacemaker. Here we have 3 waves going around. So in one geometry, we can come up with many different patterns of excitation. We were interested in describing these, and the student who worked on this, Lucas Campanari, also tried to study the ways in which such rhythms could be set up, but he did experiments in a model system. So I'm just going to show this once. There's a pacemaker over there. It sends off a sequence of waves. We form a spiral wave temporarily over here. The spiral wave keeps on going until it drifts off. We're left with waves going around the ring, and we can be left with one wave going around the ring. So if we have a circular pathway, there has to be some way to break the symmetry, and this is the way this is broken in this. Okay, now, I would like to just take a few minutes to talk a little bit. So that's all I'll talk about in cardiac system for the moment. At the meeting that was in Barcelona last year, I was delighted to meet Thomas Reed, who I believe spoke here about a week ago. So Dr. Reed has pioneered development of cytogenetic techniques to understand the dynamics of genomic alterations during the development of solid tumors. And he studied in particularly breast, cervical, and colorectal carcinomas. And I was delighted to have a bunch of conversations with Dr. Reed in which we thought about the question, well, thinking about mathematical aspects of genomic instabilities and growth stability be fruitful in cancer research. Now, I was interested in early hypotheses concerning growth control in cancer. I gave a talk at the NIH in Dr. Reed's laboratory in December of this last year, and I was a little bit surprised to learn that these ideas are completely forgotten by the cancer community. The basic idea, and this was work primarily by a scientist named Bullough, and papers published in high impact journals such as Nature, in which they demonstrated or purported to demonstrate that there could be specific chemicals that were produced by a tissue that would diffuse through the tissue and would also regulate the growth control of the tissue. So as the tissue grows, it sends off a feedback circuit which will regulate its own growth. Okay? That's the idea. That's what they were proposing. They were proposing there are chemicals that do that. So it's a negative feedback of growth. People here may or may not know that if you have some of your liver removed, the cells will grow back until you have approximately the same mass of liver. There's some feedback control. If you cut your skin, the skin will grow back until it has the same geometry. So one of the notions was what kind of feedback mechanism is regulating and controlling this geometry. In an early paper, I proposed the potential mechanism, which was just really, so this is now a reaction diffusion equation. This is from a paper I published in 1973, in which a substance, these substances were called chromes, are produced, they diffuse, they're being destroyed, and the production is just being produced where the tissue is. So one can take those parameters, and depending on those parameters, if one says that if the concentration of this is greater than a certain amount locally, then we no longer have growth. There's this negative feedback. Then one has the possibility of having an instability. So if this parameter crosses a threshold, we go and we have uncontrollable growth. That was the idea. As far as I know, it's completely wrong. There's no evidence to say it's right. I just like the idea. Okay? How could we possibly, what kinds of mutations could possibly happen? Well, what Dr. Reed is interested in is changes in the karyotype of the organization. What's the karyotype of the organization? The karyotype is the structure of the chromosomes, the organization of genes on the chromosomes, and how these genes and how these chromosomes are affected by various modulation patterns which control the expression of the genes. Okay? So the karyotype relates to chromosome structure and organization. Now, I have, I guess, a hobby of trying to understand something about karyotypic organization, because different species have different karyotypes. The chromosome structure in different species are different. Somehow the karyotype has to change over evolutionary times. Normally we say that if you have reproduction, you need to reproduce with an animal, an organism, that has the same karyotype as yours. Well, if that were true, you could never change the karyotype. So there's a question, how can the karyotype evolve in time? Once again, there were people who have thought about this going back a long time, in particular M.J.D. White, who talked about mechanisms that could lead to the change of the karyotype. I was also surprised to learn that a lot of that literature is not, apparently is lost to people who are working in this field now. Okay, so I wrote notes about how can it evolve a long time ago. And it seems to me that what's likely is that the karyotype inside the genome, there are parts of the genome which can act to rearrange, to cut it apart and paste it together. And in any one organism at any one time, the karyotype, the geometric structure of the genome, is stable under these various parts of the karyotype that act to cut it apart and splice it together. So, in other words, that in a stable species, the karyotype, the structure of the gene, the structure of the chromosomes, is a fixed point under operators which can act to cut it apart. We know that there are closely related species in which the karyotypes are wildly different, that have evolved over very short periods of evolutionary time, which would indicate to me that the evolution of the karyotype is happening by some mechanisms which are more than just simply random chance happening. I think there's something more than a Darwinian random chance plus selection. There are very powerful techniques that are used to see which parts of which chromosomes in a person are associated with which parts of which chromosomes in related organisms. These are called chromosome painting, and one of the things that Thomas Reed and colleagues have used these in related techniques to try to analyze the chromosomal rearrangements that are taking place in cancer. So, similar chromosomal rearrangements are happening in different people in similar types of cancer. The question which I pose and which I discussed with Thomas Reed is whether or not those changes that are happening may be happening randomly or whether they may be associated with particular functions and particular genes in the chromosome that that can act to rearrange the chromosome. Okay? So Thomas Reed has indicated characteristic changes in chromosome number that happen sequentially as time goes on. So it seems to me that if you observe these kinds of changes, then there's something that's happening, possibly something that's happening above and beyond just random effects. Okay? So I think this poses a number of questions related to karyotypic evolution in cancer. How does specific mitotic inhibition occur? Search for mutations in gene lines, cell lines. Different cell lines have different karyotype. How stable are those cell lines? How do mutations occur in those? Search for sequential changes in the evolution of cancers and the regulation of transcription and translation of genes. Now, my talk dealt with the possible role for nonlinear dynamics in helping the diagnosis, treatment, and prognosis of human disease. I really just focused on heart disease and cancer. I believe such an approach is relevant for other diseases of other organisms. There are many neurological diseases, diseases of motor control, respiratory diseases, in which there are really striking changes in the dynamics and organization of the dynamics of key physiological functions in the individual. So I believe that there are still challenges for the synthesis of genetic and dynamical approaches in the study of disease. I've been very fortunate of having worked with a large number of very talented students, colleagues, collaborators, and I have mentioned some and tried to acknowledge them on the slides as I've gone along. So I've also greatly benefited from funding agencies in Canada that have supported this work. So once again, thanks very much, and I'm delighted to answer questions. Yeah.
So, Professor, I would like to first to ask is that does your cardio model actually can predict how environment affect the heart operation?
The heart operation?
Yeah.
In what?
In what sense? So I'm not so sure if that your model actually include how environment actually affect those kind of like heartbeats and normal fluctuations.
Right. So most of our work has been done in model systems. When we show a record like that of a woman who died suddenly, we do not know what happened in the environment during that period of time. One of the things that I think is missing, and what's a problem in terms of doing cardiological analysis, is exactly the issue that you're raising. That one has a record of the heartbeat, one can do that, but rarely is that record correlated with what that person was doing at that period of time. Did they take medication? When did they eat? When did they exercise? I would also mention now that with wearable devices, there are possibilities for collecting vast amounts of data from mostly normal people. And there are big challenges in doing that in terms of a clinical medical study because of need for anonymity and so forth. But if you wear a Fitbit or something around your chest and you go exercising, you can post that on a website showing what your heart was doing. You can say what you ran, how far you went, and so forth. And people are in fact doing this to some extent at the moment. It is possible that we're moving into a time period when there'll be large amounts of data collected from bodily rhythms, including the heart, which will be made available for scientists, for medical doctors, for whoever wants, to do data mining and data analysis to try to understand more about causative factors. At the moment, to the best of my knowledge, this hasn't happened. I think it will happen. And for people who are entrepreneurially oriented, this is an interesting area. The wearable devices and collection of medical data using wearable devices is something which is potentially happening in the future.
We're going to take one more question.
Actually, I got one more. So it's kind of very interesting that your cryptotype, is it cryptotype?
Karyotype.
Karyotype, okay, karyotype. It seems that like only Chinese people have a very huge difference than like other region of the world. So am I getting that right? Could you turn back to the one that you're showing the different?
Oh, it wasn't Chinese people. Sorry. It was Chinese and Japanese raccoon dogs.
Oh, okay. It's
Okay? Chinese raccoon dogs had 254, and Japanese, that's the number of chromosomes, 238. I expect that if you saw a Japanese and a Chinese raccoon dog next to each other, they would look exactly the same as one another.
But then I was kind of thinking, so it seems that the different regions, the differences, I personally feel that is very huge. Is it very huge for this kind of differences?
Yeah. So this is something, you know, there are cytological journals, and I should give the reference for this. I don't have the reference on the slide. Munchak is a kind of deer. Okay? These are just examples where people notice that there are very large differences in the number of chromosomes between very related species. And because of notions of when these speciation events occurred because of geographic difference, probably, this happened in comparatively short evolutionary time. So I think it's a puzzle how these changes took place. what are the mechanisms that are regulating and controlling these changes, or are they just not being regulated and controlled, or they just happen randomly by some governing type mechanism?
I see. Thank you.
Okay, let's thank the speaker again. If you have any question, you can just ask.
Yes, so I'll be around for the next couple of days, and I'd be delighted to talk to anybody.
We will have a break for 30 minutes.